If you leave something out, you might find you can't build your knowledge. Okay, let me ask you before I start. Do you know what a polymer is?
A what?
A polymer. What's a polymer?
It's a chain of amino acids.
It's a chain of something. It's a chain. Yeah, that's good. It's a chain of things. It could be a protein is the acid. That's a polymer. But a chain of sugars, that's a polymer too. A freight train with train car, that's a polymer also. When you have repeating units of the same basic kind, that's a polymer. Now, this is an enzyme right here, and he makes polymer. So, RNA polymerase, that's the one that snaps together RNA nucleotides and make a big old long polymer. DNA polymerase does it for DNA, snaps them together and makes the long polymers. Now, this whole thing is talking about something called transcription. Let me tell you what it boils down to. I am the DNA. I truly am the boss. But I can't visit my kingdom. I can't even leave my house. I'm locked in the nucleus. So if you're all my workers out there in the cytoplasm, how do I tell y'all what I want y'all to do?
Send a message.
That's the messenger RNA. I have a messenger out of RNA I can create. And that messenger is small enough to leave the nucleus and go out there, and you're all ribosomes. And you're making different things. So transcription is how the DNA actually sends the code or the telegram out to the ribosome. And it's going to ribosome. All right, now. And to do this, to make the messenger, you use the polymerase. It's going to be a chain of acids. And you've got to unwind the DNA so you can get there and actually do the making. You only make one side. You don't make both sides. Only one side codes for maybe your eye color. But it does use that one side, and if the RNA polymerase, if it sees a C, what's it going to bring up the tie to it? What matches a C?
G.
A G. And if he sees a G, he's going to bring up a
C.
Now, if he sees a T on the DNA, he's going to bring A up. Watch out now. If he sees the A, though, what can he not bring up? They have no T's. So what does he bring up instead?
U.
The U. U is a twin of the T. I don't know why T don't work both ways, but it doesn't. I just know that when that RNA sees an A,
look for U.
He finds a U because he's making RNA. Now, if he's making DNA, then what's he going to find?
T.
The T. And that's the only difference in two of those two. C's and G's work beautifully. But when you're going DNA to RNA and you see an A there, your first thought is, give me a T. No, not a T. I'm making RNA. Give me a U. And that's how that works. Because in your mind, you can always assume, like 1 equals 1, T equals U. Okay? Now, and y'all know this from the last time I taught y'all, that C's and G's go together. And when you're working with DNA going to RNA, you use A and U. Because as I told you, this is only in DNA. But you're making RNA. You're making messenger. So I can't use T in anything but DNA. And U stands for uracil. Now, and what the polymerase does, polymerase binds the new nucleotides together to match the DNA. So if the DNA says C, it's going to bring in a G. And then if the next one says A, it's going to bring a U in because RNA has no T's. Now if it sees a T in the DNA, it's going to bring the A in because A does match T. It also matches U. What happens, too many people are trying to they'll see a T and they'll bring a U in. You can't hook A's to A's, can you? You can't hook C's to C's, can you? Well, you can't hook T's to U's either because they're the same, basically. So when people they're so concerned about, I don't want to mess up, and they mess up. They'll see T And instead of using the A like it's supposed to, their mind's so locked on T's and U's, they bring a U in to miss it. You can't hook a U to a T. No more you hook a T to a T. That don't happen. Okay? And then there's also a sequence that stops the building. So let's say, even when we write a telegram, I'm in San Francisco, stop. I'll be in Columbus by stop. They'll use stops then. But these things use stops also. There'll be a stop codon that tells the messenger, this is it. Take off. Carry your message out there. Okay? The blueprint is what the mRNA is. Yeah, have you ever seen a blueprint? Like for your house? That's what the guy uses to build your house.
A guide.
What color is the blueprint using? Blue print. Why ain't it green print? Or a red print? I don't know why they use blue for it. It could be a black print, but it's a blueprint. Alright? So this blueprint, which is the messenger you just made, goes out into the cytoplasm where you can find the workers, the ribosomes, that actually build the protein that the DNA wanted. And if you had a prokaryote, there's no nuclear membrane to worry about. So you don't need a messenger. And a prokaryote, the ribosomes just surround the DNA and do it directly. There's no middleman. You see, if you have a membrane and I can't get out, I need my middleman. But if I have no membrane, hell, I can talk to ribosome directly then. So prokaryotes, but then again though, because there's no membrane, that also causes a chance for more disruption of the nucleus too. So some folks think, well, it seems like to me that having a membrane would kind of slow you down. And it does, but it also makes sure that the DNA remains intact. UV radiation destroys cells. By having those DNA molecules inside that protective bag away from the sun, you afford protection to it. If you're floating around in a cell and you happen to float up by the window and the sun hits you, you might be altered then. And we know flu vaccine, they change every year because of that, but they do it on purpose because by the time they get a good medication for it, next year that medication won't work anymore. All right? Now, we are eukaryotes. That means we have a nucleus. We have membrane organelles. And the only way for that messenger to leave the nucleus is to go out through a hole, and they're called pores. Pores, and they go out through the pore. And they're going to go find the ribosomes. There's two places you'll find ribosomes. Floating around the cytoplasm and also glued to the ER. You'll find them two places. Now, the ones in the cytoplasm, they're making proteins the cell will use for itself. The ones on the ER, they're making proteins to transport to somewhere else. Because the ER, that's your subway system. That's your railroads. So you have ribosomes just floating around inside your cells, and they're making proteins for your cell. And you got the ribosomes that are actually stuck. We call that rough ER. Remember it had the dots all over it? Well, those ribosomes are making proteins, but those are for export. I mean, the things we make in our country, do we always use them here? We export some of them.
We do it,
We
especially.
export a lot of them, don't we? So some farmers are making corn to eat here, and some farmers are making corn to be shipped to China and places like that. So if you're a farmer making your corn to go to China, then you're on the ER. You're about the railroad. If you're making yours and you have to stand up by the highway, you drive by and pull over and get you something yourself. Okay? Now, like I said before, to make the RNA, you got to separate that DNA molecule. It cannot stay hooked together. And the 5 to 3, remember that point? And how you stick the one above you? That's still 5 to 3. Even when you're still doing 5 to 3. So that point always sticks the one above him. So 5 to 3 is what I knew I needed to cover it for DNA because you're going to see it again here. So it's better to learn it then than try to learn it here. But it does occur in the 5 to 3 direction. And the polymerase is the one that's hooking them together. And the DNA is your guide. And by what DNA has, you know what you have for RNA. Now, this is an example. I saw a number of pictures. I like this one because it tends to show pretty much what I'm doing. Now you know how these bases pair up. Now, if you see a T, then what do you know you're talking about? RNA or DNA?
DNA.
You know it's DNA. But if you see a U, then what do you know you're talking about?
RNA.
Because they don't mix. They don't mix. And the A's are in both of them, the C's and G's. So there's only T's and U's that really separate the two together. Now, this is the, as you know, the DNA strand. I see T's, right? So if you see an A, what comes in from the RNA?
U.
No, T, right? Now, if you see a T, you don't come with U now. You come with the A. If you see a C, RNA comes in with the G. And if you see a G, it comes in with a. And if you see an A, you come in with U. And you see they're coming this way. See, this is 5, this is 3, this is 5. And the 5 is going to point him. So we're building in this direction. I'm going to use the strand right there. And when it's done, it's going to leave, and then it's going to re-zip and recurl back up. But the red strand, this is what you're making. Which are the older ones? On this end or on this end? A or B? Now these are the old ones. He's the first one laid down. Because I'm still laying there. I'm still paving road over here. This road's done paved. So this thing is moving this way. And when it's done, this red strand, that's going to be called your messenger RNA. And that's going to leave the nucleus and go out somewhere and find himself a ribosome so that ribosome can read this code and make what this thing wanted. Okay? So, and this is me talking. So, that's how your genetic information gets put on RNA, by reading these base pairs. Read the base pairs. The base pair reading, that's all there is to it, really. I mean, that is critical understanding DNA if you know about A's and T's and C's and G's and U's and that kind of stuff. Okay? Now, that is transcription now. Now, replication was what? What was replication? If you make a replica, is it almost like something or just like?
It's like almost
A
like
replica?
Almost like.
It's just like it. So to do replication, I'm using DNA to make more DNA. But now transcription, I'm like translating. Well, actually, I am using DNA to make RNA. That's not replica. That's not replica, is it? I got U's and T's in there. That's not replica. So if you're doing replication and you're making DNA, well, you see an A, you can put a T down there because you're making DNA. But on transcription, you see here, if you see the A on the DNA, well, RNA comes in, there's no T's come in, right? It's got to be a U. It's got to be a U coming in. So the message that's transcribed, that's your RNA. That's your RNA. And that is used to make a protein. So that's why, who actually controls the making a protein? It ain't the messenger RNA, it's the DNA. Because the messenger got it orders from who? Now, without the messenger, would it get done though?
No.
No way. Without the messenger, you won't get anything done. But still, the messenger is not the king. He's just a courier. The king is DNA. Okay.
What is the message from
Now, what you got to remember, everything you see on DNA is not always used to make RNA. You're going to hear about things called interons I don't know if I did that or not. Some places are junk. And what happened, say your DNA repairs itself. Say you got sunburn and the DNA messed up. What's going to happen is that the other side is going to fix the broken side, but not erase the broken. There'll be a piece there that is nonsense, because here's the one that's been fixed. It's like when you mark our answer off and write a new one down. Your old one's still there, right? So, and I know grading papers, I don't check when you mark through, because I know that's not what you wanted. Well, in the case of DNA, it repairs itself. So the part it fixed, it stays there, but the new part goes next to it. So when you're looking at this transcription, you don't transcribe the bad stuff too, it's just the good stuff. So if there are 1,000 nucleic acids, every one of them is not going to be used. You may have a bad section there that got repaired. It's not clipped out. It stays there. But when you start doing your messenger, it knows to start at the good one and ignore the bad one. It might even work down good, skip the bad, and go again. So, see, a lot of people think that every single piece of DNA is usable. That's just not true, because DNA has the great ability to repair itself, but it doesn't throw out the bad piece. The messenger somehow knows that, oh, don't read this. This will get me in trouble. Just skip this and keep on reading. That's how it works. And so, as I mean here, every bit of it is not changed. Every bit of it is not changed. Because when it gets tainted, the DNA is repaired, then you ignore the taint. Now, I think I'm going to stop right there. I'm back, huh?
Is that still called the codon, where that starting stop is for the good and the bad?
The codon. Every 3 letters is a codon. That's it's called codon. And AUG is always the start codon. That's your methanome for anything. And then your stop codon is not really a stop. It doesn't code for anything. If it doesn't code for anything, then nothing goes there, and then it breaks away. If you code for something, it's going to bring it in. You're going to keep on making that protein. There are 3 stop codons. And I forgot now what they were, but there's no acid that ties to them. So when it lands there, that's how it stops the sequence. And a ribosome's got 3 sites. The arrival site, the peptide site, and the exit site. And the way the things are made is so cool. I mean, I'm teaching things now. I didn't even know when I was in school back in the 70s that we learn now. I'm going, and what I did know, what I learned now, I'm going, this is even more cool than I thought it was. I'm totally amazed when I see something go, man, that's just, and you talk about evolution. It's like somebody planned this. I mean, the more I read, I'm just totally amazed. First off, how it works. The second thing, that we're not messed up already. There's so many things go wrong in you. In fact, you're healthy, but count your blessings. Well, I'm not going to do, because see, the 3 steps, they get kind of big. So I'm on 3rd step, and it's going to end right there. But I want to take some time and not rush through this 3rd one. So I'm going to stop now.
When we come back from break,
huh?
when we come back from spring break,
yeah, we'll come back from spring break. That way I won't be half into it, and then you'll get half.